Hello, everyone. I'm Anup. I work with the Freiburg Galaxy team in the bioinformatics group at the University of Freiburg. I'm a graduate student and I did my studies in computer science. And yeah, so I'll be talking about machine learning, Galaxy, and a couple of projects that I'm involved with. I have divided my talk into three parts. First part is I will introduce machine learning and explain some basic concepts in machine learning. And then in the second part, I will be talking about how to use machine learning with Galaxy. And in the third and the last part, I will talk about a couple of projects that involve machine learning, and I'm part of those projects. Let's start with some basic concepts about machine learning. Machine learning is, as we know, it's nothing fancy. It's just a computer program that learns some rules from the data. Data is the most important part for any machine learning project, along with the algorithm that we want to use. So these algorithms, they learn these rules, and using these rules, they distinguish from one pattern to another. And these rules are nothing but mathematical functions. We use training data for training our models. And then using the trained model, we apply these trained models and predict unknown outputs of unknown sequences or unknown data. And this unknown data, we generally call it as test data, which the model has not seen before. Machine learning algorithms, they apply mathematical rules inside. Therefore, they work on numbers, and they do not work on text or characters. So if we want to apply machine learning, for example, on DNA sequences, then we should convert DNA sequences to numbers following there are many encoding techniques, for example, one-hot encoding. And using that, we can convert them to numbers and then apply our favorite machine learning algorithms. One of the tasks we can see here is the digit recognition, handwriting digit recognition. In each row, we see handwriting, handwritten digits are there. For example, row 1 is 0, row 1 is 1, and so on. Machine learning algorithms, they try to map the features, for example, all the ways in which zeros are written to its output, and its output is 0. When we give the algorithm this handwritten digit, and which is 0, then it tries to map the features which it has learned before using the training data, and then it will give the output. There are many applications of machine learning in real life. For example, face recognition, image recognition, email spam filtering. Then it's also used for predicting, for example, if somebody has a particular disease by learning on gene expression profiles. And there are so many in real life. For example, Google Translate uses deep learning to translate from one language to another. And also in the field of bioinformatics as well, there are lots of applications such as disease prediction, and then drug response prediction, and so on. There are different types of machine learning based on the kind of data that we have and the aim that we want to achieve. One of the types of machine learning is supervised learning. In this kind of learning, we use label data. Label data is, for example, we have the data for weather and then or climate, and then we want to predict temperature. Therefore, temperature becomes the output of our data. There are two parts of supervised learning: classification and regression. We will see briefly about these two types as well. Another type is unsupervised learning. In unsupervised learning, we do not have output defined. And using these algorithms for supervised learning, we find structures in the data. There are a couple of types of unsupervised learning. One is clustering, to find different groups in the data by using just the data itself and no outputs. Another type of unsupervised learning is dimensionality reduction. We will see both these types as well. The third type is reinforcement learning, which is generally used for robotics and for games as well. There's another type, is called semi-supervised learning. That's a mixture of supervised and unsupervised learning. In that kind of learning, we have a part of data which is labeled, but many data samples, they are not labeled. In practice, creating labeled data is expensive, and we have less labeled data than unlabeled data. Unlabeled data, it's always a lot. Let's look at classification. Classification is one of the types of supervised learning. As I said, it learns on labeled data. We can see a table here which defines the status of a person's health. For example, if a person is obese or normal or overweight or extremely obese, and this is the output of a person's health status. And this output can be mapped to different features of a person. For example, what is the gender of a person, what is the height and weight of a person, and what is the body mass index. These 4 columns, we can say these 4 columns as features for our algorithm, and the status column as the output of all these features. When we give this kind of data to our algorithm, then our algorithm will map these features to the output. And when a new set of features come, whose status we do not know, then the algorithm will make use of all the learned patterns from these features and try to predict the status of a person's health. Here, we have defined this column as output. Therefore, we have label data, and we can use classification algorithms on this kind of data. And these output, different types of output are our classes. For example, obesity is one class, normal person is one class, and so on. In regression, nothing changes except the output. In regression, the output is not a discrete quantity. Output becomes a real number. For example, if we are doing house price prediction, then the house price, which is a real number, I mean, it can be 100,000 euros. This is a real number. And predicting such quantity makes the task regression. Other features just stay the same. Here in this plot, we can see square feet, the area, which is one of the features of housing data, and how this feature correlates with the price of houses. One general example of pipeline for supervised learning would be, we have raw data here, and then we divide our data into training data set, and also the test data set, which become unknown observation. Then we apply our algorithm on this training data and make a model here. And using this model and the test data, which the algorithm has not seen before, we will try to predict the output. There can be different such pipelines. We can include more complicated steps, but this is the concept how a general pipeline can be created for supervised learning. There are many algorithms for doing supervised learning. The simplest one are the linear models. Linear models are faster to use and they are easy to understand, but they are not complicated. If the data is not linearly separable, then linear models will not give good results. And in real life, generally data is not linearly separable. However, it's always good to start with a simple model. For complicated models, we can use support vector machines. It can learn nonlinear boundaries and only support vectors, which are the data points closest to the boundary that it learns. And it's actually very good for binary classification problems. When we have only 2 classes in our data set, then support vector machines is recommended to use. Then we have decision trees, which learn simple decision rules from the data. For example, is my age greater than 40 or not? Then the samples which fall in the age greater than 40 will go to the right side of the tree, and the samples which fall in the category of age less than 40 fall on the left side of the tree, and so on. But decision trees has some problems. It has high variance in the output. To mitigate this problem, we use ensemble models. In ensemble models, we use many decision trees and take the output, which is the majority voting from all the decision trees. For example, if we are trying to predict a blue ball and a red ball, and we have 10 decision trees, and out of 10, 7 decision trees say the ball is blue, and only 3 of them say the ball is red, then we will go with the majority and say that the unknown ball, the color of the unknown ball is red. Then we have another complicated model is a neural network. To train neural networks, in general, we need a lot of data. We generally don't train neural networks on 100 samples or 200 samples. There is no good number for it or recommended number for it, but more is always better. When we have more data, then we go for neural networks. As we discussed, linear models, they learn linear decision boundaries. For example, in this image, we can see this red line is the decision boundary. On top, we have all the blue balls and blue dots, and on the bottom, we have all the green dots. However, in real life, this data cannot be linearly separable. We may need non-linear boundaries like a curve or so to learn the decision boundary. But linear models are easy to use, easy to understand, and they are faster. Therefore, if you have a data set, it's always good to start with linear models and then go for more complicated models. When we have non-linearities in our data sets, then we should try to use support vector machines, nearest neighbor models, then decision trees and ensemble methods. And if data set is bigger, then we should also go for neural networks. Here in this image, we can see the brown dots and the blue dots. We cannot separate them with a straight line. So the data that we have here in this plot is not linearly separable. And it happens with many data sets in real life that they are not separable with a straight line. Therefore, we need complex models which learn nonlinear boundaries in the data. Then we have unsupervised learning. In unsupervised learning, we do not have a supervision of labels. We have no classes defined here. And in real life, we have a lot of data sets which have no labels defined, because assigning labels to each data point is a costly operation. And we need to spend a lot of money and a lot of human work hours to create these labeled data sets. But unlabeled data is a lot. To find structures in the data, we can use unsupervised learning. And a couple of techniques of unsupervised learning is clustering and then dimensionality reduction and so on. In this plot, we can see on the left, we have unclustered data. And then after applying some clustering algorithm, we can see there are 3 clusters in the data, blue, green, and red. What is clustering? Clustering is automatic way of finding categories in the data. What kind of categories are present in the data? We do not know. These categories, the number of these categories also we are not sure about it, how many categories it can have. And these categories are clusters in the data. And the clusters, and in each cluster, the data points are together based on some kind of similarity. The similarity can be anything. In mathematical terms, we can say the similarity is in terms of Euclidean distance between 2 multidimensional vectors, or it can be Manhattan distance, or it can be a cosine distance, and so on. Finding clusters in the data is an iterative process. We assign clusters initially, and then over multiple iterations, we make these cluster centers better, and we specify then convergence criteria, and then we stop our algorithm. Some common clustering techniques are k-means, hierarchical clustering, and density dB scan algorithm. We can see one running example of k-means here. In this example, we have the data as green dots here, and then we assign 2 crosses here, one blue and one red. These are random initializations. We do not know where are our cluster centers. We assign these, and then in the next iteration, we find all the points which are nearest to both the cluster centers, and then we assign all the points which are nearest to the red cross as the red cluster, and same with the blue clusters. And similarly, we try to iteratively improve these cluster centers, and then at the end, we find our actual cluster centers, and the data is clustered into 2 clusters. In k-means algorithm, we need to specify the number of clusters from the beginning. But we do not know what is the ideal number of clusters. Therefore, we should try with different number of clusters in the data. K-means is a simple algorithm. It's simple to understand and to use. However, if the data sets become very large, then k-means algorithm may not work, or it may require a lot of time to finish, because the running time increases with the number of data points, because we need to compute the distances of each data point to a cluster center. One algorithm called dB scan. In dB scan, we do not need to specify the number of clusters from the beginning. It automatically finds the number of clusters. Therefore, it could be when we are not sure how many clusters we have in a data set, then we can use dB scan. Another type of unsupervised learning is dimensionality reduction. In biology, we have data sets when, for example, we have gene expression data sets, and then there are 20,000 genes, but 20,000 columns which correspond to each gene, but the number of samples, number of rows are very less, which correspond to different people. Therefore, this problem comes under the case of a curse of dimensionality. We have, say, collected data from 100 people, then we have only 100 samples, but the genes are 20,000. Then what happens? We are not able to capture the all combinations of the data that can be present in those 100 samples. And when we try to use such kind of data sets with machine learning algorithms, then it will overfit because the data itself is not generalized. And therefore, what we need to do, we need to remove such genes, for example, in this case, or such features which are not important for our prediction task. And this is actually the dimensionality reduction. We try to reduce the number of dimensions or features which are not important for our predictive task. There are a couple of examples, a couple of techniques which we can use for dimensionality reduction. One is PCA, is principal component analysis. It's a linear approach where we try to find most important dimensions and remove all the unused dimensions. However, this also suffers because this is a linear approach and may not work if there are non-linearities in the data. To solve that, we can use autoencoders. Autoencoders are neural networks which automatically provides non-linearity in the approach to reduce dimensions. We will briefly discuss about artificial neural networks as well. Artificial neural networks, these are inspired by our biological neurons that work in our body. The dendrites and axons, they carry signals in biological neurons. In the same way, the neural networks have been designed where the edges of the network carry information from one neuron to another. And this analogy has been used to create many neural networks which work on different kinds of data sets. For example, images and DNA sequences and so on. We will see a couple of networks in the following slides. In this picture here, we can see a simple neural network. As I said, it also has neurons. All these circles here are neurons. And all the edges here, they try to pass information from these neurons to forward. Therefore, this kind of neural network is called feed-forward network. The first layer here is the input layer, which interacts with our raw data, and then tries to learn complex patterns in this green section here, and tries to produce output at the right. Each such network has an architecture. Architecture comprises of different components of the network. These components can be anything. For example, each vertical is a layer. The layer which interacts with the data directly is called an input layer. Then we have hidden layers in the center, which learns complex patterns from the data and try to map to the output. And at the output layer, which is shown as red, this is the output layer where we collect our output. Output could be discrete in case of classification, and it can be a real number in case of regression. The components also include different error functions, the loss function here. There are different error functions, mean squared error, cross entropy loss, mean absolute error, and so on. There are different optimizers. We try to optimize the error between the predicted quantity and the true quantity. And these layers, hidden layers, can be of different types, depending on the type of data that we are dealing with. Recurrent layers are generally used for time-dependent data sets. Convolutional layers are generally used for images. For example, COVID CT scan images. And these activation functions are mathematical functions to transform output from each of these neurons. These are just mathematical functions which are applied to different neurons. We can see one example how we can design such a neural network using one data set. Let's suppose we have 6 different features in our data set: age, salary, education. This data set, we can assume this belongs to one employee, and this employee has different features such as age, salary, education, city, and so on. The input layer, which we saw here in the previous slide as yellow. So the number of such input or number of such neurons in the input layer depends on the number of features. If we have 6 features, then we specify 6 number of neurons in our input layer. And this one is the bias. In practice, we can actually remove it because neural network automatically understands, but that's a technical detail. In general, we specify the number of neurons in the first layer as the number of features in our data set. The hidden networks here, the hidden part of the neural network, the number of neurons or number of these verticals, these numbers are undefined. We do not know. It's for experimentation. There is no recommended number of layers or number of neurons in the layers. These are hyperparameters of the neural network, and it varies from data set to data set. The number of verticals is also important because when you increase the number of verticals here, a number of hidden layers, then the time complexity of the algorithm or the running time of the algorithm increases, and the neural network becomes more and more complex. And when it happens and our data set size is small, then there is a problem of overfitting. Therefore, one should always try to find out the ideal number of the hidden layers and the number of neurons in each hidden layer. And this technique of finding the ideal settings for a neural network is called hyperparameter optimization. This is one of the tasks when you actually deal with neural networks. On the right, the output neuron gives the output. This can be a real number or it can be a discrete class as well. Let's discuss about some general recommendations for using machine learning algorithms. In general, our data sets are not clean or not standardized. Therefore, it's very important to pre-process these data sets to look for any outliers or incorrect labels, or some features may have large values and some features have really low values. Therefore, these features need to be standardized and scaled to a similar level so that algorithms learn equally from all of these features and not just learn one feature and ignore all the rest. Sometimes for some features, we also have a problem of missing values when the data is not present for a feature. Then we can also use some imputing techniques to provide the most suitable data point for that missing value. It's one of the tasks that we do before using our actual machine learning algorithm, so that the algorithm has access to a clean, good data so that it can learn general features from the data. One of the tasks or one of the methodologies to use machine learning with data sets is to split data sets into multiple parts to imitate the real world environment. Suppose we have data set having 100 samples. Then we divide this data set into train, test, and validation. For example, we say there is also not a magic number for this division. For a general rule, we can say we divide these 100 samples, 70 for training, and then 20 for test, and 10 for validation. Using this training data set, we train our model, and we do not let our model see the test data. Test data is completely untouched and unseen by the model. And then using the validation set, we see how good we have done, how good our model has trained. And then we can use test data to imitate the real world environment when an unknown observation comes in and we do not know what would be the output. So therefore, to experience that kind of scenario, we use test data and see how good our model has learned. Another data splitting technique is k-fold cross-validation. In this, we do not divide our data into test and train. We use the whole data set and divide it into k parts. That's why it's called k-fold. It can be 5-fold cross-validation when k equal to 5. Then, for example, in case of 5-fold cross-validation, we use 4 parts, which is the 80% of all the data for training, and the 20 parts for testing, for validation. And then we we move in such a way so that we have used all such k-folds for training and testing as well. So 4 parts we use for training and 1 part for testing, and then we move on and go in a circle. These are 2 common techniques for splitting data sets. One of the important points for using machine learning is to use the right algorithm. Also, there is it's a hard problem to find the right algorithm. It really depends on the data, what kind of data we have. Sometimes the data is linearly separable, then maybe using only linear models is sufficient. But in other cases, when data is complicated and cannot be separated using a linear boundary, or the data has no labels, then we may need to use unsupervised learning. Therefore, it's always good to start with simple algorithms and then iteratively move to complex algorithms. In real life, the data is can be imbalanced. For example, in a cancer data set, maybe 99% of the samples have no cancer features, and only 1% of the sample have cancer features. This is one typical example of data imbalance. If you provide this data as such to a machine learning algorithm, then it will learn all the general features of no cancer. And predicting cancer would be really tough when we do not balance the data set in advance. In general, data is always imbalanced. We may not have equal number of samples for each classes. Therefore, we should always try to balance the data sets. There are lots of techniques for data imbalance, fixing data imbalance problems. Another good approach to find a general model is to tune the hyperparameters. In any machine learning algorithms, we will have lots of parameters. These parameters have default values set. If you use scikit-learn or TensorFlow or PyTorch, for example, the default values are set, but these default values may not be sufficient for the task at hand. Therefore, we need to find the right values for these hyperparameters. And this process is called tuning the hyperparameters or optimizing the hyperparameters for a particular data set. We should also try to look for overfitting. Overfitting happens when our algorithm tries to memorize the training data without learning the general features. We can see it when the accuracy is very good on training data and not good on test data. This is a very simple observation, what we can see. If we perform very good on trained data and not good on test data, it means that overfitting is happening. And it can happen when our data set is small and we are trying to use complicated algorithms on that. Another good approach for a good learning is to find accuracy for each class. It can also be related to data imbalance problem. As I discussed before, if we have 99% of the samples for no cancer and only 1% for cancer, then the accuracy on no cancer class would be very high compared to the 1% of cancer data sets. Therefore, it's good to evaluate accuracy for each class so that we know our algorithm is working equally well on learning features and patterns for each class in an equal manner. These are the general recommendations for using machine learning algorithms. The next part of my talk is how to do machine learning in Galaxy. You may have heard about Galaxy software, Galaxy application. I'm not sure. I will briefly introduce Galaxy as well. Galaxy is an online platform for doing scientific research, especially bioinformatics research. It's free for users. Users can register for free and can access to the hardware and also the algorithms which are present as tools in Galaxy. It is built on public infrastructure, and it has been built not by one person, but by an open source community known as Galaxy community, and it has contributors from across the globe. There are hundreds of people who contribute to Galaxy in big and small way. And Galaxy has over 2,000 different scientific tools for different scientific analyses. For example, single cells, machine learning, and assembly, and so on. There are lots of tools, for example, mapping tools and variant calling tools, and so on. Galaxy provides you at the backend lots of CPUs and GPUs where you can run your long-running programs, and also provides you storage for your data sets in Galaxy histories. This is how Galaxy looks currently. As I mentioned, Galaxy tools. On the left, you can see lots of tools in Galaxy. These are actually tool categories. If you click on each of these categories, several tools will appear. Also, we can also search for Galaxy tools here on the top left. There's also a data uploader where users can upload data sets, and these uploaded data sets are shown on the right. Using the data sets from this Galaxy history on the right and the tools here, we can run our scientific analyses. And when the analysis is finished, the resulting output will be shown in this history. And as I mentioned, this is free registration. One needs to be registered so that your data set is always available to you. If you're not registered, I think the history is lost. As I mentioned, we have lots of tools in Galaxy. There are different tool suites. One of the tool suites is for machine learning. We published a paper this year in summer for our machine learning tools. It has around 20 to 30 tools, and these tools are based on scikit-learn and TensorFlow and Keras. Scikit-learn gives classic machine learning algorithms, which generally work on smaller data sets as well. We also provide deep learning tools as well, which are built on top of TensorFlow. TensorFlow is one of the deep learning packages that come from Google. And Scikit-learn is also another community for providing standard machine learning algorithms. Both are available in Python. You can also use them separately. You can also use them in Galaxy as well. The ML tools that we have, it comprises of many classifiers, regressors, data preprocessors, lots of visualization algorithms. There are lots of hyperparameter tuners and pipeline builders, and so on. The important thing when you use such tools is to find the right tools for yourself, for your data, because there is no general rule that one particular classifier or regressor will work on all kinds of data sets. However, there are recommendations. For example, ensemble methods work pretty well on general data sets. It worked pretty well. Also, support vector machines also give very good accuracy on general data sets. However, it's always recommended to find the right algorithm for the data set at hand. In Galaxy, you can create a workflow of tools and create one unit of software which runs on the data set and gives you the output. Similarly, for machine learning tools as well, we can create such workflow. We can see one such workflow for a feed-forward neural network. Here we are importing all the data sets on the left, and then we create a deep learning model using deep learning model tool. And then the training happens in this tool. And the training may take, it depends on the data. It may take 1 hour or 2 hours, or it may take multiple days. So if you are using Galaxy, you don't need to worry about it. Whenever the training is finished, your output will be available in the data set, in your Galaxy history. The fourth tool, this does model prediction. When the trained model is returned by this particular tool, and the prediction happens with the test data, and then the last tool gives you the output as a plot. We have lots of training materials in Galaxy. There are over 200 training materials in general for different scientific analysis in Galaxy. For machine learning, we have around 10 to 15 training materials using different tools, different machine learning tools. Some tutorials, they use scikit-learn machine learning tools. For example, one such tutorial is age prediction using machine learning. It uses DNA methylation datasets and gene expression profiles for predicting age of human beings. These tutorials are actually hands-on. The dataset is included in the tutorial. You can download these datasets into Galaxy, and then you can run these tools. All these steps are nicely written and which can be followed easily. There are some tutorials for deep learning as well. You can see part 1, part 2, part 3. These are 3 different deep learning tutorials as well. Also, there is an introduction to deep learning tutorial and so on. So these tutorials are actually hands-on tutorial, and they are self-explanatory as well. From time to time, we offer events also. Training events where we go through these tutorials. An instructor is there, or multiple instructors are there, and they run through these tutorials, and you can attend these kind of events. We offer these events, and you can find it on the homepage of usegalaxy.eu. And you can learn about how to use these tools, or you can learn yourself as well using these openly accessible tutorials. The link to such tutorials are given at the bottom of this slide. Yeah, if you have any doubts or questions, you can ask now. After this, I will talk about the projects that I'm involved with. So if you have any questions, I can take such questions if there are any.
Maybe we can take them at the end, Anup.
Okay, yeah, sure. Okay, then I continue. So I will be talking about two projects which involve machine learning. And in these two projects I'm involved with, one project is developing an infrastructure for writing machine learning programs. And another project is to predict protein evolution in SARS-CoV-2 sequences using deep learning. Our group mainly provides infrastructure for researchers, and they provide algorithms and support so that other researchers can, for example, bioinformaticians can run their algorithms and analyses on Galaxy. Jupyter Lab is one popular editor. It can be used for writing Python programs or programs in R and lots of other popular languages. It provides an editor for writing programs. We have built a Galaxy Jupyter Lab, and we have integrated Jupyter Lab in Galaxy especially for doing machine learning. This Jupyter Lab is a popular editor. It's used for scientific computing, for doing data science and machine learning. And also it's very useful for if someone wants to learn programming as well. For example, Python or R, it's good to, it's a great tool actually. You don't need to install anything. All the packages are pre-installed. Users just need to write their own algorithms and functions. And it's also available on web. Therefore, also there's no need to install anything on your laptops. So we have built a special Jupyter Lab for doing machine learning tasks. This is how it looks. It runs on Galaxy. It's available on Galaxy. The link can be found at the bottom of the slide. So this particular tool is integrated as a Docker container, and this Docker container, the feature of Docker container is it's a standalone system. We do not need to install anything or do anything. You just need to download it and run it in your system, and it will give you an editor. You can write your programs. So no need to install anything. And all the packages for doing machine learning, for example, TensorFlow, scikit-learn, Matplotlib, all the visualization, data manipulation tools, everything is present inside. I can briefly discuss some features of Galaxy's Jupyter Lab. So as I mentioned, this is a Docker container. It's inheriting from a TensorFlow's notebook, which already has some packages involved for doing data science and machine learning. On top of that, we have made it possible so that it can run on the machines having GPUs as well. GPUs are used for parallelization, and it helps you to run your program faster, especially deep learning programs. We have installed different packages such as CUDA packages. When this container is downloaded on a machine having access to NVIDIA GPUs, then this automatically bridges the gap between the software and the hardware, and the program starts taking leverage on GPUs. We have integrated ONNX format for saving and sharing machine learning and deep learning models. Also, there is Git integration. We can import Jupyter notebook files directly from GitHub, and also after updating, if you want to check in those code at GitHub, this is also possible via Git integration. We have integrated Elira AI. So this allows users to create a workflow of notebooks. For example, in Galaxy, we have a workflow of Galaxy tools. Here we have workflow of notebooks. Different notebooks can be connected and can be run, executed as one unit of software. Using this editor, users can connect to Galaxy histories as well using BioBlend, and you can add dataset, create dataset in a Galaxy history, and also import datasets using the Galaxy connection. Also, there are lots of different dashboards for GPU utilization and lots of other features, small features. The Docker image for this project also works with Galaxy and also works out of Galaxy at the same image. The same image can be downloaded to any machine, not necessarily having Galaxy. It can also be downloaded and work with. This is the architecture of the project. We have a Docker container here, which opens a Jupyter Lab notebook. This notebook can be executed as such. And if you know that notebook is running for maybe 2 days or so, then we can also connect it to Galaxy, and then we can just close this notebook, and all the results, when they are available, they will be pushed back to the Galaxy history. So this particular part is currently ongoing, and once this part is finished, we plan to make it available as for users and also write a paper about it. Another such project, or not such project, but it's a different project. This is prediction of protein evolution in SARS-CoV-2 sequences using deep learning. We all know about SARS-CoV-2 and COVID-19. So in this part, we will briefly see about spike protein and amino acid substitutions or mutations. We will see what are the different clades in the evolution of viral genome, and how we can model such evolution using sequence-to-sequence learning, and what is a general adversarial network, and how does it integrate with sequence-to-sequence learning methods, and try to model the evolution of proteins here. And we will see a simple comparison of the true and generated or artificially created SARS-CoV-2 amino acid sequences, and see if we can actually predict future substitutions by learning on the sequences that we already have. Spike protein, as we know, it's one of the structural proteins in the viral genome, and its genomic position starts from around 21,000 and from 21,000 to 25,000. And there are lots of non-structural proteins here, and we have 4 structural proteins. Spike protein is it's important to look at because lots of mutations are happening in spike protein region, and this protein is responsible for the viral genome to connect to host cell, human cells, and starts its entry into the host's body. The mutations happening in the spike protein, sometimes these mutations are responsible for increased infectivity or transmissibility. For example, mutations such as a mutation from D to G at 614, it enhances the replication of the virus. Some mutations that, such as N to K at position 439, it increases the binding affinity, and also it neutralizes the effect of antibodies. There are lots of mutations which are actually not malicious, but some of those are actually malicious. Therefore, it's important to study such kind of evolution and substitutions. Here we can see this is the amino acid positions, which is generally 1273 amino acids long. And we can see the mutation frequency of such sequences, which is really high. And we can see such mutations happening in different lineages of SARS-CoV-2, different lineages. And here we can see different lineages, and these substitutions and deletions, we can see it's happening a lot compared to our other regions of the genome. Next, next strain is it's one community. I think they have created a software for creating clades in different SARS-CoV-2 genomes that we have collected so far. And it shows different points of time where the evolution has happened to different categories. These categories are clades. For example, it started from 19A and then went on to evolve to 20A and then so on. So currently we are having these clades. These clades are just categories having signature mutations at different positions in the genome. I have done experiments to model such evolution by using a pair of clades. I have created a data set. For example, the input sequence coming from clade 20A and output sequence is 20B. And try to model sequence-to-sequence learning method on this. Sequence-to-sequence learning is, we can understand in a way how we translate from one language to another. For example, on the left, we have one language, for example, say English here, and then we let our neural networks learn and predict output of this sentence in, for example, in French, for example, which is done by the decoder. This is one of the standard ways for sequence-to-sequence learning and also for language translations. Similar technique I have used for learning or modeling this evolution from one clade to another. So at this point, I have used my sequences from clade, for example, 20A, and output, my sequences come from clade 20B. And I let the neural network learn the entire edit mechanism that is happening inside. And this edit mechanism gives me at the end possible substitutions that may happen in future. This is a simple workflow of my technique. I have used a parent clade, for example, clade 20A, and its children clade, for example, 20A, and I trained a model. And after training, I generated sequences, artificial sequences, and also I have access to the real sequences, which my model has not seen. And then I compare these sequences and see how close I'm predicting with the real sequences and compare substitutions. For this, I've used generative adversarial networks. These networks are used a lot for generating images and constructing 3D models of objects and language translation. These networks have two parts. One is generator and another is discriminator. Generator for this project is this sequence-to-sequence encoder. That's the generator which generates data. And in this project, generates amino acid sequences. And the discriminator is another neural network which discriminates between the true sequences which we have seen and the generated sequences by the model, by the generator. And these two networks, they get trained together, and they make each other better while training. When the discriminator starts to fail to discriminate between the true and generated ones, then we know that our generator is so good that it's generating sequences which are almost real sequences. Then the discriminator starts failing. Then it cannot discriminate between the true and the fake ones. And then we know we have to stop training and we are good to go. We can use it for generating sequences. Here is one plot where I compare the substitutions. The top one is the substitutions of amino acids. So on the y-axis and x-axis, you can see the amino acid codes. And the plot shows the frequency of mutation from one amino acid to another. At the top, it's parent clade to true children clade. It's the frequency of mutations. And at the bottom, it's the true parent and generated children. And we can see that there is a lot of similarity in these plots, which means that the generated sequences are quite close to producing real sequences and also produce real substitutions. The Pearson correlation, we can see it's around 0.68 from these two plots. And there are lots of features which are actually comparable, which says that using the sequences that we already have, we can actually predict some substitutions that may happen in future to some extent, not completely, but to some extent. For example, in one alpha variant, one signature mutation in alpha variant is N to Y at position 501. It's one of the signature mutations. So this particular substitution we can see here in the true data set, which we can see N to Y with a significant frequency we can see here, but it's totally absent in the generated sequences, which says that not all substitutions can be predicted with this kind of model, because only sequences are not responsible for such mutations happening. There can be many different factors in nature which is happening, and we cannot model them. But there are many which can be actually predicted. This is for the children clades of 20B, and similar results I got for the children clades of 20C as well. So for both these, I trained a model using sequences from 20A to 20B. So children clades of 20C, my model has not seen before. So just using the sequences from previous clades, I'm able to predict to some extent some substitutions. So this is also one of the ongoing projects. So I think with this slide, I would like to conclude my talk. And if you have any questions, I'd be happy to discuss.